Hello friends, good evening to all. The people who are crazy enough to think they can change the world are the ones who do it. So we have to think differently. Today, let's begin with a new topic, that is compound microscope. Compound microscope. The term compound in compound microscope refers to the microscope having more than one lens, devised with a system of combinations of lenses. A compound microscope consists of two optical parts, namely the objective lens and the ocular lens. Working principle of the compound microscope. Compound microscope have a combination of lenses that enhance both magnifying power as well as the resolving power. The specimen or object that is to be examined is first mounted on a glass slide and positioned on a specimen stage between the condenser lens and the objective lens. A beam of light from the base is focused by a condenser lens on the specimen. The objective lens picks up the light transmitted by the specimen and generates a magnified image of the specimen called primary image inside the body tube. This image is again magnified by the ocular lens or the eyepiece. When higher magnification is required, the nose piece is rotated. After low power focusing to bring the object to a higher power, it is from 15x to the 45x in a line with the illuminated part of the slide. In higher power, we get 45x illuminated specimen. Occasionally, very high magnification is required. For obtaining high magnification, we have to take it under 100x, that is oil immersion objective lens. The common light microscope is also called a bright field microscope because image is produced brightly illuminated field. The image appears darker because the specimen or object is denser and somewhat opaque than the surrounding. Part of the light passing through or object is absorbed. Then the magnification of the compound microscope. In order to ascertain the total magnification when viewing an image with a compound light microscope, take the power of the objective lens, which is at 4x, 10x and 40x, and multiply it by the power of eyepiece, which is typically fixed at 10x. Therefore, a 10x eyepiece used with a 40x objective lens will produce a magnification of 400x. The naked eye can now view the specimen at magnification that is 400 times greater, and so microscopic details are revealed. Alternatively, the magnification of compound microscope can be calculated as M microscope is equal to D divided by F naught into L divided by Fe, where D is the distance of distinct image which is kept at 25 cm. L is the length of the microscope tube, F naught focal length of the objective lens, and Fe focal length of the eyepiece lens. Let's see the compound microscope parts. Eyepiece and body tube. The eyepiece is the lens through which the viewer looks at to see the specimen. It usually contains a 10x or 15x power lens. The body tube contains the eyepiece of the objective lenses, that is 15x, 45x and 100x. The objective lens and the stage clips. Objective lenses are one of the most important part of compound microscope. As I said earlier, the objective lenses have a certain degree of magnification, that is 15x, 45x and 100x. They are the closest to the specimen. A standard microscope has 3 or 4 objective lenses which range from 4x to 100x. Stage and stage clips. Stage is a place where the specimen is placed, and stage clips are the clips that hold the slide in a place. Arm and base. The arm connects the body tube to the base of the microscope. The base supports the whole microscope, and in base, the illuminator or the light source is placed. Illuminator and the stage. As I talked earlier about the stage is a flat platform where the slide is placed and the specimen is viewed. The illuminator is a light source for a microscope. A compound light microscope usually has low voltage bulbs as a illuminator. Nosepiece and aperture. Nosepiece is a rotating turret that holds objective lenses. The objective lenses has 4x to 100x magnification power. The viewer spins the nosepiece and selects different objective lenses. The aperture is the middle of the stage that allows light from the illuminator to reach the specimen. Then the condenser, iris, diaphragm and diaphragm. A condenser gathers and focuses light from the illuminator onto the specimen being viewed. Iris, diaphragm adjust the amount of light that reaches the specimen. The diaphragm has 4-hole discs placed under the stage. Each hole of different diameter. By turning it, you can vary the amount of light passing through the stage opening. Let's see the applications of the compound microscope. A compound microscope is of great use in pathology labs so as to identify disease. Various crime cases are detected and solved by drawing out human cells and examining them under the microscope in forensic laboratories. The presence or absence of minerals and the presence of metals can be identified using compound microscope. Students in schools and colleges are benefited by using compound microscope by doing the study on the selected specimen such as chemicals, etc. It helps to see and observe the microbial world of bacteria and viruses, which is otherwise invisible to the naked eyes. Let's see the advantages of the compound microscope. It is simple and convenient. A compound light microscope is relatively small, therefore it is easy to use, easy to store, easy to carry from one place to other. As it has multiple lenses, compound light microscope reveals great amount of detail in sample.
Thank you for your precious time. Have a good day. And if you have any questions,